Hello everyone, I'm Sakshee Chacharkar, a volunteer with Shivi Forensics. In this video, we'll be covering MCQs on wildlife forensic. So let's begin. Now, what is wildlife forensic? So this is the branch which uses scientific and veterinary methods to investigate crimes which involves animals, which helps to determine, confirm violation of wildlife laws, identify the animals involved, and also to determine the cause of injury or death. Now let's begin with the MCQs. First, the stable isotope analysis of animal tissues is primarily used to determine. Answer is B. Geographic origin and dietary history. Now, isotope values depend on local environment and food. These values stay preserved in tissues, which help trace where the animal lived and what it consumed. Second, the most common mitochondrial gene region which is used for wildlife species identification via DNA barcoding is? Answer is COI. COI shows clear differences between species but very little variation within the same species. Now, this makes ideal for species identification and is why it is used in the barcode of live database. Third, which of these is morphological methods for identifying wildlife species from hair? Answer is cuticle scale pattern and medullary index. Now, the different species shows distinct hair scale patterns and medullary ratios. Now, these features allow quick and non-destructive species identification without DNA analysis. Fourth, in wildlife forensic, the term CITE stands for? Answer, Convention on International Trade in Endangered Species. Now, it is an international agreement that controls the trade of endangered animals and their products. Species are placed under Appendix 1, 2, or 3 based on their level of protection. Fifth, which of the following is used for individual identification in wildlife forensic? Answer is microsatellite profiling. Now, microsatellites vary greatly between individuals of same species, making them useful for individual matching, parentage analysis, and population studies. Sixth, the primary advantage of mitochondrial DNA over nuclear DNA in degraded wildlife sample is? Answer, higher copy number per cell. Now, each cell contains many copies of mitochondrial DNA but only 2 copies of nuclear DNA. Because of this, mitochondrial DNA is more likely to remain detectable and also it can be amplified even from damaged samples. Seventh, forensic entomology in wildlife crime can help estimate? Answer, time since death. Insects reach animal remains in a predictable order and also develop at known rates. Now, studying these stages helps estimate how long the animal has been dead, which is important in poaching investigations. Eighth, the Wildlife Protection Act 1972 of India categorized species into schedules. Which schedule provides the highest level of protection? Answer, Schedule 1. Now, Schedule 1 includes the most protected species with strict bans on hunting and trade and also severe legal penalties. Now, examples include tiger, elephant, rhinoceros, and snow leopards. Ninth, environmental DNA is used in wildlife forensic too. Answer, to detect species presence from water or soil samples. Now, animals releases DNA into their surroundings through their skin cells, hair, or waste. By analyzing these traces, scientists can identify species without directly seeing or disturbing them. 10th, which technique is best suited to distinguish between closely related species where COI barcoding fails? Answer, mitochondrial control region analysis. The D-loop region changes faster than COI, so it provides finer genetic differences. Now, this helps separate closely related species or subspecies. Thank you for your attention. I hope this presentation helped you to understand the basics of wildlife forensic.